The overall goal of this protocol is to describe the functional assessment of comprehensive single-site saturation mutagenesis libraries of proteins utilizing high-throughput sequencing.
This method can help answer key questions in the fields of biochemistry and molecular evolution, such as what are the biochemical, structural, and evolutionary constraints on proteins, and how can we engineer proteins with novel functions. The main advantage of this technique is that it maximizes the number of useful sequencing reads in a whole protein saturation mutagenesis study by multiplexing library construction and sequencing.
After preparing a plate of PCR master mix according to the text protocol, use a multi-channel pipette to transfer 10 microliters from the 96-well plates containing the previously diluted sense mutagenesis primers to the cognate wells in the PCR plates. Use a plate seal to cover each PCR plate and centrifuge at 200 Gs for 2 minutes. Then transfer the PCR plates to a thermocycler and run the following program. After carrying out additional PCR and diluting the reactions 1 to 100, transfer 1 microliter from the mixed and diluted first-round mutagenic PCR products to the cognate wells of new PCR plates containing master mix. Use a plate seal to cover each PCR plate. Centrifuge the plates at approximately 200 Gs for 2 minutes. Then transfer the plates to the thermocycler and run the same program listed earlier in the video. After verifying the PCR product size,